But let's go ahead and get started. So today we're going to sort of continue from where we left off last time. Last time I talked a lot about the very smallest units of chromosome structure, which could have been a big review for a lot of you, but I talked about DNA and physical properties of DNA, and then the very basic steps of how DNA first gets structured into something larger. Just to kind of review, I'll do a little bit of review of what we talked about last time to keep it fresh in our mind. And now that we're moving on to some larger scale aspects of chromosome structure, I want to remind you again that for 50 years or more now, we've been studying this level of DNA. So physically, we know a lot about DNA and how it interacts with these proteins called histones, that I mentioned last time. But once you start getting above that level, then we're in a really more experimental and less clear area of biology. But it's kind of more exciting area of biology. I don't mean to say that we completely walking around in the dark. That's where a lot of the most exciting research is being done right now. So we're going to move on to the bottom half of this picture. But just to review, I want, can anybody remember which, you might actually remember which way is which, but I also might have reversed the pictures. So I want you to, can you look again and tell me what is the handedness of this helix? And I want everybody to be confident that you know. Because I showed you the same picture as last time, but if you reverse the picture like this, you will reverse the handedness. So don't just remember what it was last year. Remember how we do this, how we figure out, you put your hand down and then decide which way the helix is moving, and then which way your fingers is, either your right hand or your left hand that goes. So how many people think this is left-handed? Oh. Nobody agrees. Okay, do you all agree that it's right-handed?
Yeah.
Okay, yes. Yeah. That's right. I think last week this was a left-handed, but I reversed it, so now it's right-handed. And then what about this one? A little harder to tell. Can we have a volunteer to shout out which way it goes? Who can get it first? And is confident.
Left-handed.
You're right. You're correct. Okay, and then DNA, I showed the correct way because I don't want to ever show you the wrong way. So DNA is always right-handed. And let's be on the lookout for people who draw it incorrectly. All right, so DNA has its physical properties. And the first thing that DNA does, we talked about last time, is it wraps itself twice. Maybe I didn't mention that last time, but DNA always makes these two loops around this 8 proteins that are bunched together called, and these are the histone proteins. So DNA comes in, so now this is the DNA double helix. It's too small for you to see, but it makes these left-handed helical coils around the nucleosome particle. And yeah, so this is always left-handed going around these nucleosomes. And the reason it wraps around these nucleosomes is because they match the size of DNA very well, and they have a lot of positive charges around the outside of these proteins. And DNA, as you remember, is very negatively charged. So it just has a very natural affinity for DNA, and it wraps it around into these coils. So the proteins are called histone proteins, and there's 4 different kinds. And then 8 of these histone proteins get together to form one histone octamer. But then this entire thing here is called a nucleosome. And then there's about 150 bases of DNA wrapped around the nucleosome. And so from here to the next nucleosome is about 200 bases of DNA. So that's the first level. And then I also went over a little bit that these nucleosome proteins aren't just these little balls. Each of these histone proteins has a tail sticking out. The protein actually starts with this tail. So this is the the N terminus, or the beginning of the protein. They have these tails that stick out, and these act like little tags, and other proteins can come along and modify those tails by putting on or taking off different chemicals. And what do those tags do? They can make the DNA more closed and open. That's in general what they do. They act as little markers for regions of the chromosome, and they can make it expand into this euchromatin state, or they can make it contract into the heterochromatin state. So for instance, if you modify your chromosomes with methylation, shown in this, with this yellow circle, then you will make it more compact. But if you take off that methylation and instead use acetylation, which is a different kind of molecular tag, then you will open up the DNA and make it more open. And the last thing at the very small level is that DNA itself can get methylated. So cytosine residues in mammals, the cytosine base can get the extra methylation put on top of it, and that also leads to a compact state. Right. So based on modifying DNA and modifying the histone proteins, DNA can expand or contract, and that's a very important level of chromosome structure. And that's responsible, for instance, for this banding pattern that we saw earlier. So the dark areas of chromosomes are where chromosomes are compacted. So histones would be methylated, DNA would be methylated, whereas the light regions, there's still DNA there that you can't see. It's just not stained very well. But those light regions are like the light regions in this cell. It's just open and more less compact. And the words that we use to describe those two states are heterochromatin for the dark parts, and euchromatin, just ignore that, for this light part that's not very dense. So this is the nucleus of a cell, by the way. And I pointed out last time that there's a lot of heterochromatin at the very boundary of the cell nucleus, and there's still some more coming about why that should be. All right, and then I talked about the fact that our DNA is compacted into our cells, and now we're going to kind of depart and cover some new ground that we didn't get to last week. So if you remember, I said that we've got a cell with a nucleus, and then in human cell, for instance, there's 23 chromosomes and 2 of each, so there's 46 chromosomes, and all together, if you line them up, this would be, if you lined up all 46 chromosomes and stretched out all the DNA to its maximum length, you would have 2 meters of DNA. So that's a statistic that's meant to impress you with how much DNA is compacted into our nucleus, and how amazing it is that 2 meters gets down to a space of maybe 20 millionths of a meter. But really, it's kind of cheating a little bit, because DNA is never stretched out like that. And it would be impossible to stretch out our DNA to its entire length, especially when DNA is dissolved in water. It never has such a straight appearance. But in fact, DNA, like any polymer, takes on a very random kind of conformation, because the DNA, if part of the DNA at the beginning of the molecule is pointed this way, it's very unlikely that DNA in the same molecule down the line is going to be pointed the same way. In fact, each little DNA segment is independent from each other, because there's nothing forcing DNA into a straight conformation. So DNA takes on what's called a random walk configuration. And that will never be the extended length of the entire 2 meters. And we can figure out exactly how long or how big our DNA should be in this random walk configuration with a little bit of mathematics. So just by there's a quantity called the persistence length, which is kind of like the unit of the unit of distance, or the length over which the direction of the polymer becomes uncorrelated. So if this part of the DNA is pointing this way, then after 50 nanometers, you can't predict which way the next segment of DNA is going to be. So for instance, if this is an actual path of DNA, then when this direction changes to some completely different direction, this distance is about 50 nanometers. So we can say that DNA is really composed of a bunch of independent units of 50 nanometers each. And so the question becomes, how are these units of 50 nanometers going to arrange themselves if you just put them in water? So there's this equation, which is, so B is that length of 50 nanometers, which is about 147 bases of DNA. And if we take our biggest chromosome, this is chromosome 1, there's 220 million bases, 220 million AGCTs, chromosome 1. And if we divide this by how many bases are in one of these 50 nanometer units, we get about 1.5 million of those units. So you can think of chromosome 1 as not as this giant long molecule, but as 1.5 million of these tiny little segments, and each segment can have an orientation that's independent of all the other segments. And the overall size, so if we took chromosome 1 and just let it go however it wanted to be in this messed up tangle, then the typical size of that would be the square root of n, the square root of this many units, times the size of one unit. Or in other words, that would be 61 microns. So not many centimeters, but actually only a ball of about 61 microns, okay? And if you did the entire genome, so all 23 chromosomes and two copies of each chromosome, then we would get a ball of randomly oriented DNA of about 300 nanometers, 320 nanometers, microns. So this would be big enough to see, but it's so this would be much bigger than the actual nucleus of a cell. So an actual cellular nucleus on average is about 10 microns. Whereas all the DNA, if you put it in water, or if you put it in a medium that's like our cells, it would be about 320 microns. So it's not 2 meters of DNA that we really have to worry about, but it's still about 8,000 times too big to fit into the nucleus, or 20 times too wide. So it's got to fit into, this DNA has got to fit into a ball and into a sphere of about 1/20th the diameter of what it wants to be. Okay, but that's much easier than making 2 meters of DNA getting scrunched down. So the problem is not as hard as the 2 meter figure makes it out to be. Okay, so how does it achieve this? So we know that it does do it somehow. We can squeeze this ball, this 320 micron ball, down into a 20 micron nucleus. And yeah, how does that work? Well, the first level, wrapping around histones, already gets us pretty far. Here, this 50 nanometer persistence length, that should be 147 bases of DNA, is already, you can see that it's already taking up several nucleosomes, each one of which is 200 nanometers. And so if you think of this DNA as being wrapped around these nucleosomes twice, then the amount of compaction we get just by wrapping around nucleosomes is already 7-fold. So we can contract this ball of DNA already 7 times just by wrapping it around these nucleosomes. Just because of the size of each individual unit, this is 200 bases of DNA that's been already compacted by being attracted to this positively charged protein surface. Okay, so we get this 7-fold decrease from wrapping around histones. Then we only need another, if it's 20 times too big, then we only need another 2 or 3 times decrease from this other higher level of chromosome packing. Okay, so after the first level comes these other levels, which we don't know quite as much about, but they mostly involve packing these histones together very, very close, so that all the extra space in between the nucleosomes is removed. And one way this has been shown to happen in some cell types and in some conditions is by packing this elementary nucleosome fiber, where you just have DNA wrapped around nucleosomes. The width of this fiber, if you look at it in a microscope, is about 10 nanometers. So this is often called a 10 nanometer fiber, just by just referring to its width. If you add a certain amount of salt, if you change the balance of positive and negative ions and make the DNA extra sticky, you can get what's called a 30 nanometer fiber, so a much thicker fiber. These two things are shown with the exact same magnification, but you can tell that there's a lot more. Things are much thicker, and it's a lot more packed in this than it is in this. And what's thought is that these nucleosomes are wrapped in an extra level of helical coiling to make this 30 nanometer fiber. However, this was those pictures I showed you. This is not inside a cell. Okay, this isn't happening inside a cell. This is someone who's taken the DNA out of the cell and added some salt and put it in an electron microscope so you can see the packing of DNA. But it's not. It was not known for a long time whether this actually occurred inside our cells. But since it can happen very easily, it was thought, yeah, that's probably how our cells do it. But then some more recent, and so this is just by adding this magnesium, you can change this 10 nanometer fiber into this larger 30 nanometer fiber. But it actually doesn't, it doesn't seem to be the case that this happens inside cells. So people have looked with other ways of examining whether there's some kind of regular structure inside DNA. And for instance, since this 30 nanometer fiber is quite regular, it's kind of like a crystal, this regular repeating pattern, and there's ways you can detect whether you have such a pattern inside cells, and nobody has ever seen it. In fact, it's much more likely that instead of being regularly arrayed into this 30 nanometer fiber, it's really just this 10 nanometer fiber that's very irregularly arranged. So this 30 nanometer fiber idea is probably not correct, but instead there are other ways that this 10 nanometer fiber gets packaged. So we're going to go over some of those. Okay, but before we go over to what that actually looks like, I need to talk about what's going on with this higher order chromosome folding. So I've said the problem is that we need to pack DNA inside the nucleus, and that's true. Of course, all our DNA needs to fit inside our nucleus. But that's kind of the easy part. If all you wanted to do was pack it inside our nucleus, you could just condense DNA, make it really sticky, and just precipitate it into a sticky ball of DNA. But we don't just want to store our DNA. We need to be able to use it. We need to be able to access it. So it can't just be this random, messed up, compact structure like this. Instead, there has to be some sort of organization to this packing. And mostly, genes need to be accessible to be turned on. I'm not really going to go too much into the molecular biology of what happens, what genes are and how they work, but just to review, we all know genes code for proteins, and in order to read a gene, some other proteins need to go bind to that gene and read the information that's stored inside that gene. And if the DNA is too packed and too condensed, then these proteins won't be able to access it. So in order for a gene to turn on, DNA has to open up, and that's achieved, as we saw, through this modification of histones and DNA methylation. All right, and the reason we know this, one of the main ways that we know that this is what DNA has to do, is by looking at a very particular kind of chromosome. These are they're called polytene chromosomes, and they're really, really huge. These chromosomes are 1,000 times bigger than our normal human chromosomes. They're found in a very limited number of species and cell types, but just fortunately, one of the most well-studied organisms, the fruit fly Drosophila melanogaster, or Shoujobai, as you may know, not all their cells, but particularly in some salivary gland cells in the fly's mouth, they happen to have these giant chromosomes. It's thought perhaps the reason is that these salivary gland cells they themselves are really big, and they have to secrete a lot of proteins to digest what the flies want to eat. And because these cells are so big and they need to make a lot of protein, they replicate their chromosomes many, many, many times. Normally we just replicate our chromosomes once, and then we divide the cells, and then replicate again, and then divide the cells. But in order to make these salivary gland cells really huge, what flies do, they replicate their DNA 10 times in a row. So here's the first replication, and then each of these DNA strands replicates again, and then each of these DNA strands replicates again, okay? And it keeps going and going and going 10 times. And after that, does anybody know how many DNA strands you will have after that process? You know the answer. What is it?
1024.
Right. Yeah. So yes, they have exactly 1024 strands of DNA, and that's why they're so big and visible. So yeah, 2 to the 10 is 1024. And those cells never divide. They replicate their DNA and then stop. Okay. And early microscopists who like to look at things and categorize things made very detailed maps of what these chromosomes look like. So because they're so big and reproducible, so the chromosomes are always the same, they could identify actual landmarks, and they named them after letters and numbers. So they made this map of these giant chromosomes they're called polytene chromosomes. And polytene just means many threads. Okay. You'll see this teen word very often, that means thread. Yeah. So just many threads are in these chromosomes. By the way, these kinds of chromosomes are in these insect salivary cells, but humans also have polyploid cells. We have some cells where we replicate our chromosomes many times. For instance, in the placenta, the developing placenta, we have cells that make giant chromosomes. The only problem is they don't stay together so regularly. So they replicate, but the chromosomes kind of move around after that. So we can't see these nice giant chromosomes in any human cells. But it's not something specific to insects. In fact, humans too have cells that behave the same way. So the striking thing about these fly chromosomes is not the fact that you get this 10 rounds of replication, because we do it too. But for some reason, after replication, the chromosomes stay right next to each other and they don't move around. And that's why we can get these really nice maps. And so now we know, they didn't know at the time, but now we know that these dark regions are heterochromatin and the white regions here are euchromatin. Just like the human metaphase chromosomes I showed you. So because people made maps and they could measure features like the size of each of these bands, they could tell when these features changed. And it was really kind of striking. So for instance, if they heated the flies, if they exposed flies to some extra heat, it was known that doing this turned on some protein expression. So if you heat up flies they will die unless they can cope with this heat response. So they have this response to heat shock, where they turn on a bunch of extra genes that help them cope with the heat. And it was found when you do this, and you look at these polytene chromosomes, some areas of these chromosomes blow up, expand a lot. Okay, so for instance, this region, this A7 and A8, A10 region, which is this region that's normally small, gets really big. So when you need to turn on genes that reside in this part of the chromosome, the chromatin actually expands. And this was one of the first indications that the structure of chromosomes actually is important for turning genes on and off. So if you need to turn these genes on, the chromosome has to get bigger in order for those genes to be turned on. Okay, so right. And it wasn't just this heat shock, but also just during normal development. As a fly develops from a small larva into an adult fly, they need to change which genes are turned on at which time. And by just looking at these chromosomes, it was found that a very predictable sequence of events happen over this course of 20 hours of development. So for instance, this 63E region. So I don't know which chromosome this is or where exactly this is, but there's a region that people called 63E that was reproducibly always looked the same. And at a certain time, this region blows up and gets really big and turns from heterochromatin into euchromatin. And much later it was found that certain genes inside that region of the chromosome, they need to be turned on at that exact time. So just by looking at these chromosomes, this did not require any high technology, and it was done maybe 50 years ago, just by people looking with microscopes at fly chromosomes. But people could understand already back then that there was this relationship between the structure of the chromosome and the developmental timing, that certain genes needed to be turned on at certain times. Okay. So we're still only talking about this level here, this level of chromosome compaction, where the spacing between these histones gets bigger or smaller, and that seems to cover a lot of what's going on with gene expression and turning on proteins. But chromosome architecture does not stop there by any means. There are many higher levels of that. So. Obviously, to get from those, to get from this kind of nucleus so where all the chromosomes in the nucleus are completely spread out. So to get from there to where? Yeah. Anyway, to get from there to here, for instance, requires a lot more organization than just opening and closing. Okay. So what happens? What happens at the next level? Where if you think of this as this the 10 nanometer fiber that has some sort of structure, and it's either open or closed. Let's say this is a more closed version. But this fiber itself has some orientation and some path through three-dimensional space. It's not just stretched out, and it's not just going randomly throughout the nucleus. It in fact has some nice coiling organization as well. And if it didn't, then that would cause problems, as you see. So DNA is not just randomly going throughout the nucleus like a bowl of udon. It's really kind of wrapped up nicely in some coils. It's not quite like this rope, though. It uses a specific kind of structure where DNA makes this axis, and then off of that axis there are these loops of DNA coming off of the axis. And that's the basic organization of DNA at a large scale. And then how do we know that this is true? Well, again, just by looking at chromosomes, at different kinds of chromosomes in different kinds of organisms. we can learn a lot about how chromosomes are organized in all organisms. Okay, so this is another kind of interesting example. Here this is not from an insect, this is actually from amphibians, from frogs or salamanders, and not from their salivary glands, but actually from the oocytes, from the egg cells. It's another case where chromosomes need to be very big, because eggs also need to be very big cells and produce a lot of protein. But instead of doing this polyteaming business, what they do instead is it's all done with the three-dimensional organization of DNA into these large loops. This is, you can't really read this here, but this is 100 microns. So this is already a tenth of a millimeter. So if you imagine, this is already it's about half of a millimeter. So you could easily see this. You could easily see these chromosomes. I mean, they're still quite tiny. But just a small magnifying glass would be enough for you to get the idea that there's some little hairs in here. And then a very simple microscope could show you something about the structure of these chromosomes. They're called lamp brush chromosomes because I guess back when they were discovered, it was very this kind of lamp was very popular. People used this kind of lamp that had fire, and then this lamp would get very dirty. And to clean this lamp, you would need a special kind of brush that was some sort of wire with a lot of hair on it like this. So we'd have to use this brush to clean the lamp all the time. And the chromosome has a very similar organization where there's an axis and then things coming off that axis. And the guy who discovered this is none other than Walter Fleming, who discovered all chromosomes in the beginning. So he also discovered this kind of interesting lamp brush chromosome. It's not very clear what's going on here with this old picture. So here's a more updated picture. They use a fluorescent dye instead of a colored dye. And it's much higher magnification. And now I think you can see that here from this axis, there are these loops of DNA coming out and coming back to the axis. So it's not, these aren't individual strands of DNA coming out, but this is one chromosome where the DNA is coming out from the axis and then coming back, and then coming out again and going in all directions. And the thought is that on each loop, there's one gene that's getting transcribed into a protein. And so these lamp brush chromosomes are probably the most transcriptionally active chromosomes known. And the reason they're so long is because it's all opened up into euchromatin. So there's no heterochromatin left, except maybe at the very middle of the chromosome where there's no transcriptional activity going on. Okay, so you get this lamp brush chromosome structure, but it's pretty difficult to imagine. How do you go from this kind of linear 10 nanometer fiber? How do you arrange this into this kind of structure? It doesn't just happen by itself. DNA just left to its own devices, DNA will always look like this. It will always make this random walk. So if you want to organize it into a higher level structure, you need to use some other proteins, just like histones. So histones are at the very basic level of organization, of chromosome organization. To go up one level, you need to have proteins that act on more DNA. So there's two kinds of proteins. In fact, now we're at the level where we're not dealing with individual proteins, but more like large machines made of many proteins. I'm not going to worry about what they're actually called, because actually in different species, they're called different names anyway. So it doesn't matter, but there's two kinds of protein machine. One is called condensin, and the other is called cohesin. So similar names, but they have quite different effects. Okay, starting with condensin. Condensin has, just like cohesin, it's drawn like this because it's made of these 5 different proteins. But for our purposes, all that really matters is that it's this kind of, it's kind of like a ring. Okay, there's long proteins that make up the sides of this frame, and then there's some proteins here that bind these proteins together. So you'll see why it's important. It's basically a lot of proteins that make this kind of open ring structure that can open and close. Okay. So condensin is required for our purposes here, mostly for chromosome compaction and for making chromosomes rigid. Whereas cohesin is required to keep replicated DNA together. So I kind of had to draw this in here. But the purpose of cohesin is after you replicate DNA, you want to keep the DNA that's replicated, you want to keep it together until it's time to divide the cell, because as you'll see, if you fail to do that, then the DNA will not get distributed correctly to daughter cells. So between condensin and cohesin, most of chromosome structure is determined. And the way it works, I hope you can see this, is you have all these little rings of condensin. Here I'm just showing condensin here, but they work pretty much the same way. They work to pinch together this 10 nanometer fiber of DNA wrapped around nucleosomes. So what it's showing here is one fiber that's going like this. It's already inside this condensin ring. And then if there's another fiber over here, then condensin can trap it and keep those two things together. So it's a very simple activity, but just with this activity, you're able to achieve a lot of effects. And in fact, just with condensin, you're able to turn DNA that wants to be random walk, that wants to be randomly organized, into something as compact and very regular as this mitotic chromosome. And if you take a close-up look at a mitotic chromosome, it's kind of difficult to tell, but each of these little knobs that you see is thought to be one loop of DNA. That's just like the white brush chromosome. Let's see. I need to come back to the next slide a bit later. Okay, yeah, so cohesin. Cohesin is not involved in making this mitotic structure other than holding the chromosomes together. So, so cohesin is involved in holding this part of the DNA to this part of the DNA. So cohesin you would expect to be located all along the center of this axis, and it's holding those two replicated chromosomes together like that. Okay. With the same sort of mechanism. And this is actually a molecular diagram where everything is shown at roughly the correct scale. So this is one DNA strand actually going around. And here's another DNA strand. And so cohesin is about as big. The hole in the cohesin molecule is just big enough to hold these two DNA strands together and keep them pinched together. Okay, and then if you don't have cohesin, this can lead to chromosome missegregation. So remember from 2 weeks ago, I talked a little bit about human chromosome disorders like trisomy, like the kind of trisomy that leads to Down syndrome, and how it gets worse with maternal age. Well, one of the reasons is that in humans especially, the oocytes that are going to turn into eggs are sitting at this stage where the two chromosomes are held together by this cohesin, and they can sit there for years and years. And as the maternal age approaches 40 years, then these molecules actually start to degrade. They just start to randomly come off. And if too many cohesin molecules come off, then the chromosomes can fall apart and missegregate. So this cohesin molecule is actually quite important, not only to keep chromosomes together during normal cell divisions, but for reproductive health, because it's responsible for a lot of the chromosome abnormalities that can occur. Okay, so these condensin and cohesin molecules, which are actually composed of many proteins, they do most of the work of holding chromosomes together. But if you think about the mechanism that I showed you, where you have one strand of DNA and then cohesin can grab another strand of DNA and hold them together. And if this is happening at a large scale with millions and millions of megabases of DNA, you might expect that there would be a problem, right? If you have a bunch of wires like this guy and they're held together randomly along their lengths, you could get this really massive tanglement, right? So why don't chromosomes get tangled together inside the nucleus? If you think about this, where of course there's lots of condensins condensing DNA here, but eventually it leads to so this kind of, all the DNA in this nucleus eventually gets resolved into little packages like this. But in doing that, why don't things get tangled together? So that's been a very interesting question, and people have not really understood the answer until recently. Potential models that could avoid entanglement have been thought of over the years, but very recently there's been some experimental evidence for one of them. So I'll share that with you right now. And it's a way that condensing molecules can get regulated to avoid entanglement. And the model that achieves this is called the loop extrusion model. So loops are the same, it's referring to the same loops I showed you, lamp-brush chromosomes, for instance. And extrusion just means just pulling out, right? Something is extruded if you pull it out. So somehow loops are getting pulled out. So we have to go to kind of a zoomed out view of DNA. So starting DNA double helix. This is something that acts on the 10 nanometer fiber. And you just have to imagine that there's this kind of molecular machine where two of these condensins or cohesins are held together in a kind of figure 8. And what you can do with this is if you pass the DNA through in this way, so it's passing in one, and then, so it's passing through two of them basically. So you have two of these condensins that are held together. Then what you can do is you can start to pull a loop of DNA through the holes, right? So this was just kind of a theoretical model for a long time, and the idea was that you don't have one condensin molecule and then another condensin molecule and then two strands of DNA are brought together like this. Because if you did this randomly, you would just eventually tangle up all the chromosomes together. Instead, you first link some condensin molecules together and then pull a loop out. And if you do it that way, you can see that you would never put any other strand of DNA through this one. So you'll have a lot less entanglement. I mean, you could have one loop like this, and another loop gets inside that loop. So it doesn't completely get rid of the problem of entanglement, but it makes it much less. Okay. And one of the main pieces of evidence for this model is that if you look really closely at these at what kind of DNA is bound to these condensin proteins, you find a common sequence at many of them. Okay, you find this a particular DNA sequence that's known to bind to a certain protein called CTCF. And so the idea is that this loop can get pulled out, and it keeps getting pulled out until a certain sequence comes in, and then it stops. So this protein CTCF that binds to this sequence is able to stop the loop from being pulled out, and that's how the loops can have normal size. And then there's a group that put out this paper where they actually did some molecular modeling. They did some computer simulation of simulating the kind of forces and kind of thermal fluctuations of the DNA fiber that would happen inside a cell. And using this model, they were able to actually get well, it's not exactly proof, but they have very good evidence that this model is happening. So DNA can be pulled through, but then if this CTCF motif happens to enter this ring, then the DNA loop stops. And I should freeze this because it's important to note here that the rest of the DNA here still follows this random walk pattern. So if there's no condensin holding this DNA together, then it's just going to be this random walk pattern. But they did some really nice simulations on not just a little bit of chromosome, but the entire nucleus, and showed that if you have this model of loop extrusion, then you can fit all these chromosomes inside a nucleus, and they will never get tangled up. So even though this looks like these chromosomes are tangled like a bowl of ramen, this red chromosome, for instance, it's never actually entangled with the blue part of the chromosome. So you can see that they keep separate domains throughout the entire cell cycle. Okay, we're going to stop that for now. So this is the basic way that chromosomes can be organized inside the nucleus without tangling with each other. Is that not only do you have these chromosomes organized in the 10 nanometer fiber around nucleosomes, you also open and close that 10 nanometer fiber based on whether you want to express the genes or not. And then you also further organize that fiber in these giant loops. And typical sizes of these loops, by the way, are in hundreds of kilobases. So if there's, let's see, if there's 5 nucleosomes in 1 kilobase, then this is about 1,000 nucleosomes worth. So 1,000 of these beads on a string in one of these big loops. And so you eventually can get an entire chromosome organized in this looped out manner. And the base of this loop always has a particular sequence on it. And this is how, for instance, the proteins condensin and cohesin are able to organize this into an axis. Because if you have condensin and cohesin molecules that bind to this sequence, those can all kind of come together and make this make the axis part. In other words, so you have these loops coming off, and this is all made of DNA. But you have also a lot of proteins that bind down here at the base of these loops. And it's these proteins that make up the axis of the chromosome. So, this is the next one here. So, right. So, the quality here is not very good. But for instance, if you look at where cohesin is on the chromosome, here the red is DNA, and the cohesin, you can see, is pretty much right in the middle. So, it's in the perfect place that it needs to be to hold these two DNA molecules together. And condensin, which is really responsible for holding these loops together, is also where you'd expect it to be. So, in other words, if you have a replicated chromosome, you'll have condensin here holding these loops, holding the loops together, and then you'll have cohesin here in the middle, holding those two replicated chromosomes together. So if you look with immunostaining, that's exactly where you see them. And so by changing the relative amounts and changing the loop sizes, you can also change the size of the chromosomes. So it's not exactly known yet still how chromosomes can be exactly the right length. So this chromosome, let's say this is chromosome 1. I don't know if it is or not. But if you look at chromosome 1, it's always the same length and always the same width. And it's still not known, so it's still kind of an open area of research how the width and length is precisely controlled. But it's known at least that it has to do with regulating the size of these loops. And so how much condensin and how much cohesin you have on the chromosome will determine the size of those loops. Okay. So with experiments where people could artificially vary the amount. So here, I hope you can see, it's green and red, which sometimes is problematic, but here condensin is here in red on the outside, and cohesin is where it belongs here on the inside, holding replicated chromosomes together. And you can see if you lose some cohesin, you will make these loops much bigger. So the same amount of DNA can be either stretched out or condensed, or even really more condensed, depending on how much condensin and cohesin you have. So the actual mechanisms, though, are still kind of poorly understood, and there may be some sort of feedback that senses how big loops are and prevents them from getting too big. This is kind of what we know now about the hierarchy. So we can fit our DNA in a useful way, in a way that it can still be active, all that DNA into our nucleus with a series of foldings. So the properties of DNA itself, the negative charges on the outside and its tendency to coil, are what allows it to bind around histone proteins. Okay, and then histone proteins themselves have particular spacing and size that helps compact DNA. That can be affected by putting molecular tags on the histone tails. This here again, some textbooks show this 30 nanometer fiber. We don't think that's really happening. We think we can skip this stage and go directly to this sort of looping stage, where this 10 nanometer fiber is made into these large loops that eventually, somehow, we don't exactly know how, forms the typical chromosome shape. Okay, so that's kind of how chromatin gets folded inside the nucleus. And then, so the next part of the large-scale structure of chromosomes has less to do, well, I guess, okay, how should I introduce this? So this folding that I've talked about is a way of getting an entire chromosome packaged and the right size. But there are other aspects of chromosome structure that have to do more with, that are more special than just this folding. You'll see what I mean. So I'm talking now about just eukaryotic chromosomes. Okay, bacterial chromosomes, like I showed you earlier, they also probably have some kind of loop structure, but they're very, very simple. And usually bacteria only have one big chromosome that gets replicated, whereas we have many, many chromosomes. And if you look, you can see that there's some structure to these chromosomes, right? You have these arms, and then you have this little bit in the middle, right? So they have a certain shape to them, as opposed to bacterial chromosomes, which are all kind of random. And you can see these are a different kind of chromosomes. These are chromosomes during meiosis. But you can see that, for instance, the ends of these chromosomes have something at them. So that, and it's been appreciated that the ends of chromosomes and the middle of chromosomes are slightly different than the rest of the chromosome. So the last part of this class will be talking about the specialized parts of eukaryotic chromosomes. So we'll start with the center part, okay? And it's not always at the center, but it's called the centromere because in most human chromosomes, it's at the center of the chromosome. But that's kind of a misnomer because it's not always at the centromere. So its role is not to be just at the center of the chromosome, but it has a certain function, which is during cell division, the centromere recruits certain other proteins, which make a structure called the kinetochore. And the kinetochore serves as a handle on a chromosome that the cell uses to pull the chromosomes apart during cell division. So, do you know the word in Japanese, dogentai? Yeah. So, I think centromere and kinetochore, maybe both dogentai in Japanese. They're actually different. Centromere refers to the DNA itself, and kinetochore are the proteins that bind on the centromere. And in both cases, it's little gang type. So it's the place where movement, the place where movement can occur on a chromosome. And depending on the size of the chromosome, this centromere and kinetochore also, of course, are very different. So, for instance, if we're talking about yeast, they usually have very small chromosomes, and budding yeast actually have extremely tiny kinetochores. In fact, as small as you can possibly get. It's just a single nucleosome, so it's not really shown very well, but imagine this is just one nucleosome. So DNA will come here and wrap around this twice, and that's it. So it's just a single nucleosome in yeast is able to serve as the handle that a cell can pull on the chromosome to pull it apart during cell division. And before I get too far ahead of myself, I should just say that what the cell uses to pull a chromosome apart is something called a microtubule. So the cell has these structures called microtubules. Yeah, that's orange. That can pull, they can exert force. So when the cell starts to divide, the chromosome is attached to these microtubules. And so it splits in half and then starts moving. And then eventually you have a cell that contains just one chromosome. And here you have another cell that contains this chromosome. So can't really animate this, but imagine this chromosome is being pulled into this cell by this microtubule. And the microtubule doesn't pull on the entire chromosome, it only pulls on a small part of the chromosome, and that's the centromere. Okay? And here this is an electron micrograph showing the microtubule going into this pile of junk, which isn't very informative, but these proteins are the same proteins that are bound to the chromosome in a cell. So this is yeast, but chromosomes like ours have of course, we're much bigger, so we need a much larger kinetochore. And we don't just use one microtubule to pull on our chromosomes. We have to use many, many microtubules because one microtubule would not be able to pull with enough force. So here you can see this really dark stuff is the DNA, and it's organized in a way that recruits these lines here. It's like this plate that gets put against the DNA. And this is the kinetochore, and then the microtubules are here so pulling on the kinetochore. So somehow this DNA is by itself is able to make this kinetochore happen. And I won't get too much into it, but the reason this DNA is able to do it is because instead of using the normal histone proteins, there's actually a different histone protein at the centromere. And that's how the centromeres are able to make this kinetochore structure. But we're not going to get too much into molecular details of the centromere. Let's stay at the big chromosome level. And I'm going to talk not about how centromeres work, but how they're useful for classifying human chromosomes. These centromeres are almost always visible as a constriction in the chromosome. So centromere DNA has a different structure, and it's always a little bit smaller, and that's why you get this stereotypical picture of a pinched chromosome, because it's pinched because the loop sizes are smaller at the centromere, and they have a different organization than the rest of the chromosome. And we can classify human chromosomes by the position of the centromere along the entire chromosome. Most of our chromosomes are called metacentric, which just means that the centromere is actually at the center of the chromosome. Okay, so this is a kind of canonical centromere, and both, it's called chromosome arms. So here's the centromere, and then you have different arms coming away from the centromere. You have a lot of chromosomes are called submetacentric, where you have one clear longer arm and one clear shorter arm. And whenever you have a long and short arm, I guess the short arm is always called P, and the long arm is called Q. I guess it's pretty confusing because the letters are so similar, and it's kind of hard to hard to tell sometimes when you're reading about a bunch of P and Q arms. But anyway, so there's a P and a Q, and the Q is the long arm and the P is the short arm. Even metacentric chromosomes have a P and a Q, but it's not always clear that one is long and one is short. That's why they're called P and Q and not long and short.
The P is for petite in French.
Sorry?
The P is for petite in French.
Is that right?
And then Q is just the letter after it.
Oh my goodness. Okay. Well, okay. I never knew that that's where it came from. Really? It's just petite.
Yeah.
Yeah. All right. Fine. Okay. Thank you. It was always a mystery to me. I thought they just picked two random letters that were symmetric. Yeah. Interesting. All right. So there's a petite arm and a long arm. And then I guess this is an ultra petite one. So acrocentric chromosomes are where the centromere is almost all the way to one end, but not quite. So there's like a little bit of a micro arm. And it actually goes even farther than that. Acrocentric is as far as human chromosomes go, where the centromere is almost at one end. But in many mouse species and many other species as well, the centromere the centromere is all the way at one end. So there are no known genes on the other end of the centromere. So they really only have one arm. And here, for example, this is chromosome ends are detected with a technique we'll talk about later called FISH. So the red dots here are the chromosome ends, and wherever you have a bright blue concentration, this is indicative of where the centromere position is in mice. And mouse centromeres are almost always heterochromatic, and that's why they show up as this bright blue stain. Okay, so these are called telocentric chromosomes, which means the centromere is right at the end. There's another kind of centromere that some organisms have that I'll talk about because it's, I could have made that a little bit bigger, sorry. So small. Called holocentric, and this is as opposed to monocentric chromosomes. So we have one kinetochore on each of our chromosomes. And if you think about it, you really should only have one because you only want one handle for the cell to pull chromosomes apart at cell division. If you had two handles, then the cell might pull them in opposite directions on the same chromosome, and you would rip the chromosome in half. So we never want to have two centromeres on one chromosome. That's actually kind of a hallmark of, you can get genetic problems if you accidentally make another centromere on one chromosome and make a dicentric chromosome, you will easily rip the chromosome in half. That happens in cancer quite often. But there are some organisms that don't mind having centromeres all over their chromosomes. And one of the organisms that I study, C. elegans, it's a small worm, is like that. So instead of having just a single centromere, they have centromere sequence. In fact, all of their DNA is centromeres. You can say the entire chromosome is a centromere. So then, why don't they have this problem that I just talked about? Why doesn't, if you think of this as one centromere, why doesn't the cell pull this part in one direction and this part in the other direction? And the answer is that when it's time to divide the cells, they condense these chromosomes so much that there's actually no room for this centromere to be pointing in opposite directions. So these chromosomes are still quite long and twisty. So the centromeres could point in opposite directions. There we go. So some centromeres could be pointing this way, and some centromeres could be pointing this way. But by the time it comes to divide, the chromosome is so condensed that all the centromeres can only point in one direction. So since they have small chromosomes, they don't have to worry about chromosomes being pulled apart in multiple directions. But as soon as you have a genome that's large enough, with chromosomes that are as big as humans, then that becomes a problem. So we and other organisms with large genomes only have a single centromere. Okay. Yeah. So since I've made a point to point out that there's these proteins called cohesins that are holding chromosomes together until the point of cell division. And they keep the sister chromatids together. And in fact, they also keep the Drosophila polytene chromosomes together. Cohesins are also involved in keeping them together. But at some point, the cell has to divide, so the cohesins have to get removed, right? So there must be some timing where cohesins are present, they're holding chromosomes together, but then they have to know when is the right time to remove them. And we're going to talk about that in the next class. How does a cell coordinate this timing of pulling chromosomes apart at the right time? So we'll finish the end of this class appropriately with the end of the chromosome. There's two main specialized parts of eukaryotic chromosomes. So I talked about centromeres, but also there's these parts called telomeres. And telo is end, and so it's just the end of the chromosome. And those, in fact, are the red dots that I showed you earlier. So on these mouse chromosomes, you can see each chromosome has a pair of telomeres at the end, because these chromosomes are replicated. So each telomere you see is a single chromosome's end, and these are two chromosomes held together by a cohesin. That's why you see two. The reason chromosomes have telomeres, there's something special about the ends of chromosomes. What's special about them is due to a quirk of eukaryotic chromosome replication. You may have had this in high school biology, I think, or you may have learned this at some point. And that is when DNA is replicating. So imagine this is one double helix of DNA, and it's getting unzipped and turned into two double helices. So last time I told you that DNA is anti-parallel and it has this 5' and 3' direction. It turns out that certain proteins can bind to DNA and they prefer to go in one direction and not another direction. And DNA replication is sensitive to the direction of DNA. It can only proceed from this 5' direction to this 3' direction. So when you unzip DNA and start to replicate it, it's very easy to replicate this part of the DNA because you're going from the 5' direction in the 3' direction. So this strand is replicated very quickly without any problem. But on the opposite strand, since DNA is anti-parallel, it can't just go with the direction of unzipping the DNA. In fact, it has to replicate this way. So every time the DNA unwinds a little bit, it's able to replicate a little bit more. Then it has to wait for the DNA to unwind again, and then it replicates a little bit more. And so it just replicates one little fragment at a time. And these are called Okazaki fragments for the guy who discovered them. But this is no problem because it makes these little fragments, and then these fragments get sealed together, so the entire chromosome can replicate. And that works really well for most of the chromosome until you get to the very end. Okay, so you can replicate one way. One way is no problem, but when you get to the end on the lagging strand, on the strand where you need to make these Okazaki fragments, you get to a point where you're not able to. The very end is left unreplicated because this gap here cannot be filled in from this direction. What it wants to do is fill in from this direction, but there's no DNA here to copy, to fill in. So if there were no special way to replicate the end, then each time the chromosome replicated, it would lose this little bit of DNA. And so chromosomes would get shorter and shorter if there were not some way of replicating the ends of chromosomes. And as soon as people understood how DNA replication worked, this end problem was realized. And so people realized that there must be something special about replication at the ends of chromosomes. And so the way this problem is circumvented in eukaryotic cells is by using, again, a special protein. There's a protein called telomerase, which is able to help DNA replication at the ends of chromosomes by bringing along its own little bit of RNA to serve as a template just to help that last Okazaki fragment get replicated. So this protein brings along a little piece of RNA that's always the same piece of RNA, and using this RNA, it's able to add a little bit more DNA to the very end, which allows this last little bit of Okazaki fragment to get filled in. So it makes this it makes the end of the DNA long enough so that it can get filled in from the other way and complete the DNA replication. But because it's the same RNA, so this template molecule that telomerase brings is exactly the same every time. So it can only extend the end of the chromosome with the same sequence. And so it repeats the same sequence over and over. Okay, so yeah, that's just showing the last step that I didn't show you. So eventually it's long enough, and then DNA polymerase can come back the other way and complete the gap. So here, for instance, is the beginning of chromosome 2 in C. elegans, and you don't have to memorize this entire DNA sequence. In fact, you don't even have to look at it. I'm just going to show you. You don't have to look at the individual letters. I'm just going to quickly go back between chromosome 2 and chromosome 3, and I hope you can tell that, of course, they're different chromosomes, so most of the letters change, except at the very, very end, which has the exact same sequence. And it's a repeating sequence that goes CCT-AAG, 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 over and over and over again, because that's the sequence of RNA that the telomerase molecule, telomerase enzyme brings to the chromosome to help it replicate. So the exact number of repeats can vary because in each cell division, maybe telomerase will extend by 2 or 3 repeats, or maybe sometimes it extends by more, sometimes by less. But overall, it keeps the end of DNA, it keeps the end of the chromosome long enough so that it can replicate, and so chromosomes don't get shorter and shorter. And so we don't really have to get into this. So this telomeres are not just a double helix. They actually do some really interesting. There's some interesting structure at the very end. Let's see if we can figure this out. So there's a bit of single-stranded DNA, and then it kind of loops back around and makes this weird loop structure. Basically, it wants to avoid an actual end. Because if you have just an end of DNA, like the end of a DNA double helix, that it kind of looks to the cell like that's a broken chromosome. Because if you break a chromosome normally, it will just have this end of DNA with nothing continuing. So in order for this to be recognized, to not be recognized as DNA damage, it makes this little loop. And only telomeric DNA will do this. And if you delete this telomerase enzyme, or if you cut off the ends of chromosomes with radiation, then you can see it really is a disaster because it does get recognized then as a broken chromosome or a broken DNA end, and you will get things like chromosome fusions, where if you break 2 chromosomes, they will get fused together, or you can get these little ring chromosomes that get, I think this is probably a ring chromosome, that gets, in order to remove chromosome ends. So the cell really does not like there to be free ends of DNA sitting around, because that's what happens when a chromosome breaks, and so it does whatever it can to prevent that. So if you remove telomerase, you will get these aberrant chromosome formations. Okay, so that's kind of the end. Yeah, I think we're going to stop there. So, so we talked about chromosome folding, and this example here is kind of back to the very, very smallest level of DNA structure. So this doesn't have any nucleosomes in it. In fact, there's different proteins besides histone proteins that are responsible for this looping. So depending on where you are in the chromosome, there will be different proteins involved in structuring the DNA in different ways. But for the most part, the centromeres and the telomeres are the only really special parts of the chromosome. There are a few others that we might get to. But for now, I think that's it, and we're going to pick up next time with talking in a lot more detail about the cell division and how cells coordinate chromosome replication and removal of cohesion between chromosomes with the rest of the events that have to happen in cell division. And hopefully I'll remember to bring enough, 1, 2, 3, 4, I'll bring maybe 6 or 7 pairs of 3D glasses. This is actually a 3D, red-blue 3D image of cells, and I will have some of those 3D glasses for you to see these images better. Anyway, that's something to look forward to next. And it's already May, next week, May 2nd. So that will be the theme of next class. So about 15 minutes early, so if anyone wants to ask any questions now or after people leave, that's fine. Yeah.
Would you upload slides to class?
Oh, right. Yes, yeah. Okay, I'll do it. Yeah.
Thank
I
you.
have now 3 lectures worth of slides, so yes. I probably have 1 or 2 copyrighted images from earlier that I had to delete because they're really strict about putting that. So I just need to edit that a little bit, but mostly I can just upload it. So yeah, thanks for reminding me. Yeah, is there anything else about the class itself, or like registration, or what about level of English? Is it too fast for some people, or is it too easy?
Very fast.
Very fast. All right. So probably having slides would help with that. You can review after. Okay. I can try to slow down a little bit. I think I just start talking very fast sometimes, so I'll try to slow down. Yeah,
Thank you.
Okay. Yeah, I guess that's all for today then. So I'll try to put those lecture notes up hopefully sometime this week.